We're going to take a look at the bacterial cell wall. So the bacterial cell wall is located just outside the bacterial cell membrane, and almost all bacteria do have a cell wall. There are a few that don't though. So the bacterial cell wall has a couple of functions. One is that it helps maintain the shape of the bacteria. And then another really important function is that it helps protect the bacteria from something called osmotic lysis. And what this is that when the bacteria are out in the environment, oftentimes they are living in a hypotonic solution. Remember, hypotonic, hypo meaning less than, so it just is saying you have less solute particles outside the cell than you do inside the cell. And that means that water is going to move by osmosis. And the easiest way to remember this is that water likes to follow the solutes. So it likes to go wherever there's more solutes. So it likes to go where the party is. So water would move into the cell, and the cell is going to swell, and if enough water moves in, it would burst and pop, and that would be osmotic lysis. So to prevent this from happening, that's why the bacteria have a cell wall outside their plasma membrane. Oops, that's a badly drawn cell wall, but you get the idea. The cell wall is this semi-rigid structure that will push back on all that water trying to come into the cell, and then the cell will not burst and you won't get that osmotic lysis. Okay, and then we'll talk about later how the cell wall can be pathogenic. Remember that word pathogenicity. Gen means to produce or create, and pathos is disease. So the cell wall can actually cause disease and make us sick, and that's especially true in the gram-negative cell wall. So we'll talk about that later. Okay, and then based on the structure of the cell wall, that will make the gram-positive bacteria stain purple, and that will make the gram-negative bacteria stain a red to pink color. Okay, and then just another term here, we've got cell envelope, and the envelope is just what is enveloping or wrapping around the cell, and it includes the plasma membrane, which is right here, and then the cell wall, and then anything else that's outside the cell wall. So in this picture, there's also a capsule, there's some fimbriae, flagella, there's a pilus, so any of those would also be part of the cell envelope as well. So it's just the plasma membrane, cell wall, and anything else that's beyond that. So that's the cell envelope. Okay, here we've got a nice picture that shows the difference between the gram-positive bacteria on the left and gram-negative bacteria on the right. So both of them have a plasma membrane right here. And then we've got the cell wall made out of peptidoglycan in blue. And you can see the gram-positive bacteria have a very thick peptidoglycan cell wall. And then compare that to the gram-negative bacteria on the right here, and the peptidoglycan cell wall is much thinner. Another big difference is that the gram-negative bacteria outside their cell wall, they have this outer membrane. And we do not have an outer membrane at all on the gram-positive bacteria, so that's really different. Another term you might hear is that peptidoglycan is also called murine. Murine just means wall, and that's where that name came from. I won't use that term. We'll just call it peptidoglycan, but you might see that in other places. Okay, let's take a look at the structure of peptidoglycan that makes up the bacterial cell wall. So it's made out of two organic molecules. So if you look at the word peptidoglycan, peptido is referring to protein and glycan is referring to carbohydrate or sugar. So peptidoglycan is just made out of protein and sugars or carbohydrates. So here's what the peptidoglycan subunit looks like, this whole thing over here. So I would start this in your notes. I do want you to know what peptidoglycan is made out of. So you've got two sugars called NAM and NAG. You do not need to memorize what NAM and NAG stand for. I will not ask you those chemical names, but just know the two sugars are called NAM and NAG. And then coming off one of the sugars, you've got 4 different amino acids. So that's the protein part. And I'm not going to ask you which sugar has the amino acids hanging off them. I won't ask you that. Okay, but just know that peptidoglycan is composed of 2 sugars, NAM and NAG, and 4 amino acids. Okay, so I would know that. All right, and then what we're going to do is we're just going to take these peptidoglycan subunits and join those together to make a peptidoglycan strand. And then we will take these peptidoglycan strands and then join them together at the amino acid little side chains here. Okay, and then where these amino acid side chains are hanging off, that's where the peptidoglycan strands get joined together. And there's different ways to join those together, which you don't need to memorize, but you can just look at those. And then the other thing up here on the top right, there's a picture where they've dissolved everything on the bacteria except for the cell wall. So you can see the cell wall is like this mesh bag of peptidoglycan. And you can see up close that the peptidoglycan cell wall is not a solid structure. There's little holes in it. Okay, so things can pass through it, and so it's not totally solid. All right, let's take a look at these gram-positive cell wall first, and we'll take a look at a close-up. So on the bottom, we've got the plasma membrane, and then you can see the thick peptidoglycan cell wall, and then these purple structures that are going through the peptidoglycan cell wall, that's called teichoic acid. Okay, and the teichoic acids, there's a chemical representation over here on the right. The teichoic acids have a negative charge on them. Okay, and that's what gives the gram-positive bacterial cell its negative charge. So all cells have a negative charge, including ours, and what gives the gram-positive bacteria their negative charge would be these teichoic acids. Okay, and then we also have this periplasmic space. So this is the space between the plasma membrane and the peptidoglycan cell wall. And it's very small in the gram-positive bacteria. So there's not much going on in the periplasmic space because it's so small. All right, let's take a look at the gram-negative cell walls. And this, so we've got our plasma membrane on the bottom. And then right above that, we've got our peptidoglycan cell wall, but notice that it's very thin, much thinner than our gram-positive peptidoglycan cell wall. And then outside of the cell wall, we've got our outer membrane. So remember that there was no outer membrane in our gram-positive bacteria. So this is pretty different with the gram-negative bacteria. And then from our plasma membrane up to our outer membrane, this is our periplasmic space. So our periplasmic space is much wider than what we had in our gram-positive bacteria. And because it's much wider space, there are things going on in this periplasmic space. So there are some enzymes in here, maybe breaking down some of the food that's transported in through the outer membrane before it gets transported across the plasma membrane. So there's stuff going on in this periplasmic space in the gram-negative bacteria. Okay, so I would definitely be able to recognize if I show you a picture of being able to identify, is this a gram-positive bacterial cell wall or is it gram-negative bacterial cell wall? Okay, now let's take a closer look at this outer membrane in our gram-negative bacteria. So this structure here on the outside of the outer membrane, you kind of got this little red piece and then this little stringy thing sticking out. That's called lipopolysaccharide. Okay, so let's take a closer look at that. So we don't have any teichoic acids, but instead we've got these lipopolysaccharides. Okay, so that's going to give us our negative charge, and that'll make the gram-negative bacteria have the negative charge, just like the gram-positive bacteria have a negative charge. So instead of teichoic acids, they have these lipopolysaccharides, and that's in the outer membrane. Okay, so these lipopolysaccharides in the outer membrane, here's a close-up view of that. And if we look at the word lipopolysaccharides, what two types of organic molecules are they made out of? And you can see that lipo refers to lipids, and then polysaccharides is referring to sugars or carbohydrates. Okay, so lipopolysaccharides are just made out of lipids and sugars, or lipids and carbohydrates. Okay, so our lipopolysaccharide has three parts. We've got lipid A, which is going to anchor the lipopolysaccharide into the outer membrane. And the lipid A is actually toxic, and that can actually make you sick. So we'll come back to that in just a little bit. And then on top of the lipid A, we have sticking out this core polysaccharide. And then at the very tip, we have the O polysaccharide, also called the O antigen. So the O antigen, or the O polysaccharide, is what our immune system sees and knows that, oh, our cells don't have that. And so we actually will react to that and produce antibodies against it. So anything that our immune system reacts to and produces antibodies against is called an antigen. So that's how it got the name O antigen. Sometimes you will see the O antigen as part of a name of a bacteria. So like E. coli 0157H7, which is a species of E. coli that lives in the intestines of cattle. It doesn't make them sick, but if we eat that from undercooked meat or maybe some contaminated water we ingest, that can make us sick. So the O in the name is referring to the O antigen that's in the lipopolysaccharide in the outer membrane of these gram-negative E. coli bacteria. Okay, so the importance of the lipopolysaccharide in the outer membrane. We talked about how it gives the negative charge to the gram-negative bacteria. So they don't have the teichoic acids like the gram-positive do. They have the lipopolysaccharide instead to give them their negative charge. And it does help protect them from being phagocytized because our cells also have a negative charge, and like charges repel each other. So if our cells, our phagocytes, have a negative charge and they're trying to grab onto these bacteria that also have a negative charge, it's going to be hard for them to grab onto them because the like charges repel each other. The O antigen also can evade the immune system because the bacteria can change the O antigen so then it looks different, and the antibodies that we produce to bind to it now no longer bind, and then our immune system has to start all over and produce new antibodies to bind to the new version of the O antigen. And so that wastes time, and the bacteria can take advantage of that and make us sick. And then the outer membrane is also important because it creates a barrier in keeping a lot of antibiotics from even entering into the gram-negative cell. So if the antibiotic can't get in to the bacterial cell, it can't kill the bacteria. So in general, start this in your notes, gram-negative bacteria are going to be harder to treat with antibiotics than the gram-positive bacteria, and it's because of that outer membrane. Okay, so the outer membrane will keep some of the antibiotics from getting into the bacterial cell, that gram-negative bacterial cell. And so in general, the gram-negative bacteria are going to be harder to treat with antibiotics than the gram-positive bacteria because of that outer membrane. And then we did talk about how that lipid A, part of the lipopolysaccharide, is actually a toxin. It's called an endotoxin, which we'll talk about later in the semester. But if you have a large number of these gram-negative bacteria in your body and they start to die and disintegrate and they release this lipid A, that can actually make you pretty sick. So we'll come back and talk about that later. Okay, and then the outer membrane actually is more permeable than the plasma membrane because it has these porin proteins in it. So it makes it pretty easy for the bacteria to get their food in, and then they can digest it in this periplasmic space, make it into smaller pieces, and then transport it across the plasma membrane to get it inside the bacterial cell. Okay, and then we've talked about how the gram-positive and gram-negative bacteria stain different colors because of the differences in the structure of their cell wall. So the gram-positive bacteria, remember they have a thick peptidoglycan cell wall. And so in that third step of the gram stain, when you add the alcohol to decolorize the cells, that thick peptidoglycan cell wall will just dehydrate and kind of shrink down and trap the crystal violet on the gram-positive bacterial cells, and they will stay purple. So that's why the gram-positive bacteria stain purple. But the gram-negative bacteria are different because they have a thin peptidoglycan cell wall. And so when you put the alcohol in them to decolorize, because the cell wall is so thin, the crystal violet just gets washed away. And so then they turn clear. And so then when you add the safranin in step 4, that red dye, then they will stain the red to pink after that. Okay, and then just some atypical prokaryotic cell walls. We have Mycoplasma pneumoniae. Mycoplasma pneumoniae is very different because this bacteria does not have a cell wall. Most bacteria do, and this one does not. So remember that the cell wall helps maintain the shape of the bacterium because they don't have a cell wall. They end up varying in shape. So they don't have a regular bacillus shape or coccus shape or spiral shape. They can really vary in shape because there's no cell wall. The other thing is that these bacteria do cause pneumonia. Obviously, you can see that from their name. And they cannot be seen with a light microscope. This is an electron microscope view. They can only be seen with the electron microscope because they're the smallest known bacteria, so very tiny. We also have our acid-fast bacteria. So remember that these bacteria have a waxy layer on top of their cell wall made out of mycolic acid that the gram stain can't penetrate through, and so you can't gram stain them, so they had to come up with a different way to stain them. So they did this acid-fast stain, and so the acid-fast bacteria would be the Mycobacterium. So remember we talked about Mycobacterium tuberculosis and Mycobacterium leprae. That's what we use the acid-fast stain to see those. And then the archaea, most of them have cell walls like the bacteria, but it's not made out of peptidoglycan. It's also made out of proteins and carbohydrates, but they're just different proteins and carbohydrates. And so it's made out of something called pseudomurine. Okay, and then this is just looking at the acid-fast bacterium. We have some Mycobacterium here. You can see the acid-fast stain that they've done over here on the right. And remember with the acid-fast stain, the acid-fast bacteria stain red. And here you can see the mycolic acids, so that waxy layer that prevents them from gram staining. But if they were able to be gram stained, would they be gram positive or gram negative? And if you look at the structure, if they were able to be gram stained, these would be gram positive bacteria. But they don't gram stain because they've got that waxy layer, that mycolic acid on top of their cell wall, so you have to acid-fast stain them instead. Okay, and then the cell wall is protective. We talked about the osmotic lysis before. Prevents the water from moving in, and that would happen in a hypotonic solution, right? So if we had our bacterial cell and it's in a hypotonic solution, water likes to follow the solutes, and so water is going to go wherever there's more solutes, and so it would go into the cell. So by having an intact cell wall, that will prevent too much water from moving into the cell, and then the cell will not burst or have osmotic lysis occur. But we can damage the cell wall with an enzyme called lysozyme that's actually produced in all of our mucous membranes in our bodies, so like in our mouth, GI tract, in our tears, anywhere you have mucous membranes, you'll be making this lysozyme enzyme, and you can see that it actually breaks apart the peptidoglycan between the two sugars, and that'll weaken the cell wall. And if the cell wall is damaged or weakened somehow, the water will just move in and then the cell will burst. And then the other thing that will also cause problems is certain antibiotics will inhibit cell wall synthesis. So when the bacteria divide, if they're not able to make new cell wall with these new bacteria that have been made, then water will just move in and the cell will burst. And if you want to watch this video here, it's pretty cool. If you just kind of focus on the bacteria growing on the left side of the screen, they've been exposed to penicillin and you will see them popping. It's pretty cool because as they divided and they made new bacteria, they weren't able to form a cell wall. And so the water just moves in and they burst, which is pretty cool. Okay, and then here's just a table that compares the differences between the gram-positive and gram-negative cell walls. We didn't talk about everything here, so if we didn't talk about it, you don't have to know it. Okay, and lastly, I just want to compare and contrast the differences between the gram-positive and gram-negative bacteria and their cell envelope. And I don't know why the color is not showing up, but think you can go ahead and still see this. Okay, so some differences. And this would be really good to know for the test. So I definitely would put a big star on this. So for gram-positive bacteria, they have a thick peptidoglycan cell wall. And gram-negative bacteria have a thin peptidoglycan cell wall. And then another difference, gram-positive bacteria, there's no outer membrane. But the gram-negative bacteria do have an outer membrane. And then the gram-positive bacteria have a small periplasmic space. And the gram-negative bacteria have a much wider or larger periplasmic space. Oops. Okay, let's just say space. Okay, and then what gives them the negative charge? The gram-positive bacteria, the teichoic acids, give them their negative charge. And then in the gram-negative bacteria, it's the lipoic acids, or sorry, the lipopolysaccharides. Or LPS for short, that gives them their negative charge.